SANBI Biobank is based at the National Zoological Gardens campus in Pretoria. Our collection is from close to 85,000 individual animals and storing approximately 190,000 various samples from room temperature to minus 196 degrees Celsius. The individuals represent 221 families from nearly 750 genera with almost 1,000 species represented.
Welcome to the Biobank.
Enjoy the tour. These species include mammals, birds, reptiles, amphibians, and more recently, marine and freshwater invertebrates. They are collected from 44 different countries and include IUCN Red List of Species categories extinct, extinct in the wild, critically endangered and endangered, near threatened, and vulnerable. The sample reception laboratory is considered the only dirty area of the bank and is where all the biomaterials are processed for moving into the various banks. The freeze-drying lab lyophilizes samples for future resuspension and extraction and is just one of the ways samples are stored. The room temperature tissue bank stores various biomaterials in a controlled environment. These include hair and feathers, samples collected and curated for the Google Barcode of Wildlife project, blood on FTA paper, as well as the biggest veterinary histopathology formalin-fixed paraffin-embedded wax block collection. These are used for retrospective studies. The Biobank is an access-controlled environment, with access into the banks allowed by Biobank staff only. All fridges and freezers and ambient temperatures of the banks are monitored 24 hours a day. We have two DNA banks. DNA Bank 2 are for samples currently being sorted and catalogued, with provision made for fridge, freezer, or ultra-low temperature storage. The herpetology collection previously banked at Kirstenbosch is now curated at the Biobank, and this includes 17,500 samples of reptiles and amphibia. DNA Bank 1 is where samples are stored at ultra-low temperatures ranging from minus 80 to minus 150 degrees Celsius. Fibroblast samples are stored in a dormant live state and include the extinct western black rhino, 6 northern white rhino cell lines, as well as many other species. Blood, tissue, and DNA extractions at minus 80 degrees Celsius are stored in 2 ml cryotubes, and exact locations of each tube, including freezer, section, rack, box, and site, is captured on the database. The dispatch lab is where we prepare sampling kits, as well as some of the media prep. The cryobank consists of 18,000 plus semen and biopsy straws submerged under liquid nitrogen, which is topped up weekly to maintain the collection. The collection is also stored submerged rather than in a vapor phase due to the temperature requirements of both semen and biopsies for cell culture. At these cryo temperatures, these samples are considered alive. In order to make the best use of space within the tanks, they are stored in straws, both the semen and the biopsy straws. Currently, the walk-in fridge houses our ethanol samples, and the walk-in freezer or the frozen post-mortem samples. The tissue culture laboratory is where biopsies from animals are grown into fibroblasts, which are renewable cell lines. These cells are grown in controlled environments, harvested, and frozen. Very exciting is the carrier typing system, which are in the process of procuring through the BBSA, which will be used for looking at the animal's chromosomes. And lastly, we have the pathology bank situated outside due to the formalin samples posing a risk to humans. Each individual animal has a hard copy data sheet capturing all the metadata, including taxonomy, identification, collecting events, and the biomaterials, which is then stored on our database, Specify. And lastly, the team. Portia Choma, Katune Shunzi, Kenilwe Musupi, and Gaugelo Matloa, and I'm Kim Labashane. Thank you.